Recording started.
Hi Bio30 students, it's Mr. K coming back live to you, well almost live on this recording. Hope you guys have been doing really well. Hope you had a really good spring break and that your exams went well as well. Hope you've been enjoying the warm weather. It's been really awesome. I've been really enjoying doing rollerblading with the kids, just bought some new rollerblades. So anyways, wanting to get into this stuff here, and this is going to be preparation for our test next week on Tuesday, which is the, just let me check here, 27th, April 27th. So this is going to be on lessons 7, 8, and 9. And once again, if you know the material from this review, that should help you a lot on the test. So let's get into the stuff then. First question says, somatic cells are the cells of an organism except the... Okay, so let's just read over the material below, and I'll let you guys come to the conclusion as to what the answer should be. So the info below says, remember that somatic cells are the regular body cells. So we could call those 2N or diploid cells. And sex cells, on the other hand, are the cells, the other kind of cells we find in the body, and they're 1N or haploid cells, and we also call them gametes, right? Okay, so based on that info, which ones do you think are not somatic cells in this question here? Okay, so let's go on to the next one. You'll find that in this session here, we won't have as many heavy-duty genetics questions, you know, kind of analyzing the genetics of it, as we did before. So I think you'll be relieved with that. So the next question says, artificially cutting out any gene using restriction enzymes and moving it to any chromosome. And it's asking you which process that's going to be. Okay, so we're moving a gene, we're cutting it out, and we're moving it to a chromosome. Well, I've got a bit of an example here. And you can see here we're cutting the genes of this using this restriction enzyme. It's called ECOR1. So we're cutting these genes and taking just a part of these genes. And the same with this piece of DNA. We're cutting just some of the genes out. And then we're combining these two genes together. And so first of all, we use this enzyme called the restriction enzyme to cut. And then we join together the pieces of DNA using another type of enzyme called a ligase. Okay, but this whole thing of taking the DNA, usually from one creature, and putting it into another creature is called genetic engineering. And so one of the main applications for genetic engineering today is in agriculture, where we're breeding new types of corn or new kind of animals that are more hardy, that sort of thing. Okay, so genetic engineering is when we take one part of the DNA of one organism, add it to the DNA of another organism. So that's one process we're looking at. Another process is gene therapy. And with gene therapy, we're either adding or taking away genes. I would say most of the time we're taking away genes with gene therapy. And the purpose of gene therapy is to treat a disease. So instead of just creating some new breed of corn, which is going to be hardier, gene therapy is trying to treat a disease. Gene mapping, on the other hand, is finding out where genes are found on DNA, in other words, the chromosome of a creature or an organism. And so remember, this is done by finding the percent recombination that occurs. All right, you might remember that we looked at the different percentages, and then using those percentages, we can put together a prediction of where the genes are on that chromosome or on that piece of DNA. And we did that in one of our lessons. Okay, so that's gene mapping. So using those definitions, read over this question and decide which one of these processes you think it's going to be. So again, artificially cutting out any gene using restriction enzymes, moving it to a chromosome. Which process do you think that is? And again, if you don't have time to answer the question as we're going along, later on when you look at the review sheet or this practice sheet, then you can do it at that point. Okay, let's go on to the next one. So actually this is just showing, I was talking about gene mapping, and this is showing. So it's showing how each one of these bands kind of represents a gene that's on this chromosome. Okay. The next question here says, a permanent change in a gene usually resulting in a recessive allele, which could even be lethal. Okay, so is that recombination? Is that mutation? Is that a wild-type trait? Is that a bar body? Well, let's just look at a few definitions here. Recombination is the process that occurs in sexual reproduction that results in new varieties of genes being produced. So you think about the word recombination, recombining, right? And again, remember that this process occurs in meiosis. And again, meiosis is a type of sexual reproduction of cells, whereas mitosis is asexual. Okay, so that's recombination. And then mutations, on the other hand, are permanent changes that occur within genes. Most often, mutations are problems and lead to a creature having poor health or even death. But with simple creatures, like a bacteria, a mutation could lead to it becoming better able to adapt to medicine so it can no longer be controlled by them. And you guys might have heard of this before. You know, they have certain types of viruses or bacteria which have mutated, and because of that, our medicines are no longer able to control them. So again, when you're looking at a bacteria, it's one thing. A mutation, which is normally something that's dangerous, could even result in death. The bacteria, it could be useful because it can help it develop a new trait that makes it better able to adapt to the new environment. More complex creatures, mutations rarely, if ever, lead to something better happening when they occur. Okay, so wild type allele. Okay, so this is just showing you an example of a mutation, and you can see here that we used to have a G here, and that was changed to an A when a mutation occurred. Okay? So again, remembering what these letters stand for, these different bases. A is adenine, G is guanine. Okay, so we're getting a mutation that results in guanine being produced, or sorry, adenine being produced rather than the original guanine. Okay, so that's a mutation. Okay, so you can look at those definitions there and decide what you think the answer would be for this question. Good stuff. So, on to the next one. The next question says, a sweet pea plant variant was found that has purple flowers. When this plant was self-pollinated, the following F1 offspring were produced. Remember, F1 is just the kids that are produced from the parents. F2 would be the grandkids, you could say, from the original parents. Okay, so we notice that we have pink flowers, purple flowers, and burgundy flowers. And the question says, which pattern of inheritance for flower color is demonstrated by the sweet pea variant? Okay, so it's either going to be X-linked recessive, autosomal dominant, or incomplete dominance. So let's review each one of those types of patterns of inheritance to decide which one this one likely is. Okay, so first one, autosomal dominant, means the trait we're talking about is dominant. So you only need to receive one dominant gene, and you'll have the trait, whether it's a disease or whatever. And if you check out the chart on page 612, you'll see an example of this pattern. Then there's autosomal recessive, which means the trait we're talking about is recessive. So in this case, rather than having to receive both, or one gene, you're going to have to receive both genes, and then you'll have the trait. Okay. So if you look at the chart on page 613, that's an example of autosomal recessive. Now, incomplete dominance is when neither gene is dominant. So instead, if you have a copy of both genes, you get traits that are halfway in between the two other traits. So for example, if the two other traits were black and white, you wouldn't get black being dominant over white or vice versa. You'd get gray. Okay, so that's incomplete dominance. And X-linked traits, you remember back to hemophilia, they're covered, or carry, I should say, on the mom's X chromosome. And males are more affected with these traits, and you remember what the reason is? It's because the female can have one of the recessive genes handed to her. So for instance, if it's hemophilia, she's got an X little r, but as long as she's got an X big R, then she's that trait is covered, or that gene is covered by the dominant gene, the normal gene. Whereas with the male, as long as he gets that recessive X little r, or X little h, I should say, then he's going to get the hemophilia. Okay, so again, you're going to see males getting the trait more often when it's an X-linked trait. So again, read over those again and decide which type of inheritance pattern is going on for this question. So going on to the next one, this one talks about snapdragons, so a type of flower. This says snapdragons may be tall or short, they may be red, white, or pink. The alleles for short is recessive and the alleles for color display incomplete dominance. A plant heterozygous for both traits was self-pollinated and produced 200 offspring. How many of these 200 offspring would be expected to be both tall and pink? Okay, so there's some important things we need to take note of here. The plant is heterozygous for both traits. Okay, so both color and height. And then also we're looking at how many of the 200 are going to be both tall and pink. Okay, so we know first of all that we're looking at incomplete dominance as far as the color goes, right? Because we know that the color can be red, white, or pink, and pink is in between red and white. So again, looking at an example of what incomplete dominance looks like, we see here's the gene for red, here's the gene for white, and so if you've got both these CRs, you're red, like this one is. You've got both the Ws, you're white, but if you've got a R and a W, then you're pink, which is halfway in between. Okay, so that shows you how you can get the pink flowers. Okay, so now that we know how we do the genetics for the incomplete dominance, let's look at how many of the 200 are going to be both, what was it again? Both tall and pink. So, both tall and pink. Let's underline that again. Okay, so if they're tall, again, these are the genes we're going to have, just defining the genes. That's what they're going to look like. So, if they're both tall and pink, okay, so both parents are tall and pink, they're going to have these genes. Okay, they're heterozygous for both traits. Again, heterozygous, what does that mean? It means you've got one of each allele. Okay, so they're heterozygous for tallness and they're heterozygous for color. Okay, they're both going to be heterozygous. And then we cross them. Well, if we're going to find the chance of being tall and pink, you might remember when we discussed this on our last session, the chance of two independent events is when we multiply the two events together. Like if you roll a dice and flip a coin, you multiply 1/6 times 1/2. And it's the same deal here. So the chance of being tall, you just focus on the T's here. And so these are all the 4 different possibilities you'll get if you just focus on the T's. And so knowing that being tall is when you have the dominant gene, the dominant allele, what do you think the chance of being tall would be? Again, we want this as a decimal eventually because we're going to multiply these two decimals together to get our answer. Okay, so I'll let you guys decide what the chance of being tall is based on these results. Now, what's the chance of being pink? Well, we know that these are our answers we're going to get. So if you use the parent's genes, again, the heterozygous genes, and you cross them, well, pink flowers are going to have one R and one W. And we can see 2 chances of that. So I'll let you decide then what that will be as a fraction, the probability of being pink, and then what that's going to be as a decimal. And again, once you know the probability of being tall and the probability of being pink, then you multiply those 2 probabilities together to find the chance of being tall and pink. Okay, and that's all it's saying on this next page here. Très bien, mesdames et messieurs. Okay, so next question here says, in a cross between a red bull and a white cow, a roan calf was produced. When the roan offspring was crossed, the resulting offspring appeared in the following ratio: 1 red, 2 roan, 1 white. The type of inheritance here is, okay, so this is a term we haven't talked about so much, a roan calf. And let's check that out, what that means. So again, as we talked about earlier, with incomplete dominance we see red and white flowers, and the ones that have both the R and the W. So for instance, excuse me, CR and CW genes, they would be pink. So they're midway between red and white. The difference between incomplete dominance and codominance, such as you see in shorthorn cattle, like the one we see below, is rather than having traits that are in between white and red, we see that the cattle have both white and red in them. So they're not pink, instead they're both white and red at the same time. So that's what a roan cattle means. Both the red and the white are dominant, so because of that we call it codominance. Okay, so now recognizing what these terms mean, you can look at the question here and decide what kind of dominance is going on. Alrighty. So going on to the next question, it says the ratio between the different phenotypes in a dihybrid cross should be. Okay, so we got 4 different possibilities. And how are we going to decide what it is? Well, first of all, we need to know what a dihybrid cross is. So what does dihybrid mean? Well, first of all, what does hybrid mean? Hybrid means you've got one allele of each parent. And so if it's dihybrid, it means you're talking about 2 different traits, and each one of them is a hybrid. In other words, it's kind of like what we were looking at in an earlier question when we talked about heterozygous for both traits, for tall and for, what was it, color, I guess? I think that was the snapdragons. So this is the same kind of a deal. Okay, so when we're talking about a dihybrid cross then, remember a monohybrid cross is when 2 purebreds with different traits cross to produce a hybrid. Okay, but a dihybrid is when, okay, sorry, this question is a bit different than the other one I was talking about, because a dihybrid refers to when you have 2 purebreds being crossed with different traits. So, sorry, I just got to do something on my email. I'll be right back. There we go. Okay, so that's what a dihybrid cross is. So remember that. That's important to remember. It's when you have 2 hybrids, or sorry, 2 purebreds being crossed. Okay? Now, if you check this out then, these are 2 purebreds being crossed. So this one is a purebred, and it's got the dominant trait for both traits, dominant genes for both traits. This one is also purebred. It's got the recessive genes for both traits. That's what a dihybrid cross is. Okay, so when we do this cross, first of all, you notice where you get the alleles from, right? So for this parent here, you can get some possible alleles, and the other parent you get some possible alleles. And when you do the cross, these are all the different possibilities you can get. Okay, but you can read through this here. I'll let you guys read through this section here, and you can then decide what the ratio will be, and what the ratio is that you can see there, that's the ratio you're going to have for any type of dihybrid cross. Okay? So again, just read through that section there. Okay, next question. Fertilization which occurs during sexual reproduction is more advantageous than budding because, okay, why is it more advantageous than budding? Well, we talked earlier in the session about how recombination is a process that occurs during meiosis. During this process, new variations of a gene or of gene combinations are produced. This is one of the reasons why sexual reproduction has advantages over asexual reproduction, especially if there's a change in the environment. Okay, because if there's a change in the environment, you want to have variability. You want to have, within your population, different types of creatures, and that's what sexual reproduction gives you. Okay, so with that information, you'll be able to answer this question. And going on to the next one. Okay, the next question says, use the following info to answer the next 2 questions. Okay, so we've got the name of a, I'm not even going to say it here, it's a rare genetic disease that causes problems with most embryonic ectoderm development, does not usually affect the nervous system. Individuals are born apparently healthy but lack sweat glands. So we've got a pedigree chart like the ones we've seen before, and it asks us for the inheritance pattern. It gives us 4 different possibilities. Okay, so to look at which possibility it likely is, let's look at some info here. So again, remember that when you're looking at pedigree charts, males are the square boxes, circles are female. Shaded means you're afflicted with whatever the disease is, or it might not be a disease, it might just be a trait. Okay, so every organism, every individual who's shaded has the trait that we talked about in this question. Okay, so with inheritance patterns that follow normal dominance recessive patterns, males and females have the disease in equal amounts. This is not true of sex-linked or X-linked diseases, where the disease is passed through the X chromosome of the female. Females may be carrier of the disease without showing it because they carry the recessive gene, while if males carry the recessive gene, they will show it. Okay, so because of the fact that males are going to show it more often than females, what type of inheritance pattern do you think this suggests? Okay, so we'll move on. Next question says, many adult newts and salamanders have a remarkable ability to regenerate amputated limbs. That's really cool, right? I mean, it says here you can amputate a foot of a newt and it will regenerate the lost foot. Very unique, very cool. However, if a newt has its foot amputated and receives a particular dose of vitamin A, the animal grows back a whole forelimb, not just the foot. So, a logical interpretation that can be drawn from this information is that vitamin A may play a major role in growth by... Alright, so we're talking something having to do with growth. You know, you've got the regrowth of a limb. So are we dealing with mitosis or meiosis, and are we increasing or decreasing it? Well, let's look at the difference between these processes again. So, I just asked some questions. What's the difference between the two, and how are they different in terms of how the parent cell compares to the offspring cell? That question is probably not as important as the first one to answer this question. Okay, so meiosis again is sexual reproduction where gametes are formed with half the DNA of the parent cell. Mitosis, on the other hand, is not for sexual reproduction, it's for growth and cell repair. Both the parent cells and offspring cells have the same amount of DNA, and so both are going to be 2N, whereas with meiosis, the kids are going to be N. But focus on what the purpose of meiosis and mitosis is, and that will help you to answer this question. So which one of these processes would be involved in the regeneration of limbs with newts and salamanders? Okay. On to the next one. Okay, so this... sorry about that. I just put that out of the room. Okay, back. So this diagram here is just showing us meiosis, I should say, because as you can see, the daughter cells have only half the amount of DNA as the parent cell. Whereas if they had 2, representing 2 chromosomes, then it would be mitosis. Okay. So on to the next one. It says polyploids are organisms with 2 or 3 or more complete sets of chromosomes. Okay, so normally in humans we're talking about diploid organisms, which are 2N, but you can also have 3N organisms and 4N. So 3N is triploid, 4N is tetraploid. So all major groups of seed plants have some polyploid members. Plant polyploids are larger than plants with 2N chromosome number. So plant polyploids with even number chromosomes 4N can usually produce ploin and seeds. Plant polyploids, okay, that's a bit of a tongue twister there, with odd chromosome numbers, example 5N, are nearly always sterile. So if we have a 2N plant, a diploid plant, and a tetraploid plant, each capable of normal meiosis, if we're crossing them, the chromosome number of their offspring is going to be somewhere between the two. Okay, so I'll let you decide what number is going to be between the two. And then again, remember that diploid is 2N and sex cells are going to be haploid or N. That's not really important for this question. Okay? So, going on to the next one. Okay, so this info here is just info to know. It's not really to answer a question. The farther apart genes are from each other on a chromosome, the greater the chance that a crossover may occur between those linked genes. Okay, so this is showing crossing over, and also we're talking here about recombination. Occurs during meiosis, produces variability since some genes from the mom and some from dad are being joined on the same DNA. Okay, so again, not important for the question we're looking at. This is our last question here. And it says, in a genetic cross, we have these genes being crossed with these genes to produce 4 types of gametes in unequal ratio. Gametes capital A, small b, and A, capital B would not be possible without crossing over. Of 300 offspring, 9 had the genotype here, these 4, and 9 had the genotype seen here. Okay, so these particular individuals, these 9 here and these 9 here, are going to be what we call recombinants. Okay? They only could have occurred if recombination could have happened or had happened. So we call those the recombinants, these 9 and these 9. Okay, so it says find the percent crossing over. Well, to find the percent crossing over, we need to use this formula. So we find the individuals, the number of individuals that represent recombinations. We divide that by the number of offspring given in the question, and then we use this here, this fraction, to get a decimal. We multiply that decimal by 100, and that's going to give us the percent, for the percent crossing over. Okay, so you always need to know that formula there. Alrighty, so it looks like we're done in less than half an hour here, which is good. So again, I encourage you guys to watch this before the test, which is going to be next Tuesday. And again, we have more tests coming up as well. We have one on DNA, one on populations, and then after that we're at our final exam and of course the diploma exam. And so I'd encourage you guys to keep going, pedal to the metal. This is a really important time to be keeping up in the assignments and doing well on your tests. And so I want to wish you guys the best on your test next week. And God bless. Have a great day. Take care.